So good morning. I start recording the first section of today's webinar. Good morning to everybody. As you already know, this webinar are organized for the iPlanta program, and we record the webinar. All the speakers agree to have the webinar recorded and also agree to publish their presentation on the iPlanta web pages. Vera will chair the session. If you have questions, please remember to, during the presentation, to post your question on the chat, and then Vera will manage to transfer your question to the speaker. Then after, we will have a general discussion. I want to thank Vera and all the speakers for their availability to organize and make this first section happening. I think that it's really interesting to have this first exchange with different stakeholders and see what can be discussed in the future of the RNA technologies that we have developing in the iPlanta program. I stop here my presentation, my very short introduction, and I give the floor to Vera. Thank you, Vera.
Yes, good morning. I'm Vera Ventura. It's really a pleasure for me as the chair of the Working Group 4 on Socioeconomic Impact to open this event. Working Group 4 deals with the analysis of socioeconomic aspects of RNAi solution, but also on creating networks with stakeholders. We organized a similar event in 2018 in Brussels, and today we want to repeat the experience to open again the discussion on RNAi and the potential use of these new plants and products. So the event is structured in 2 sessions. This morning we will meet an open discussion with stakeholders, and we will look at the challenges of modern agriculture from the producer's perspective and how RNAi technology can contribute in providing solutions for these challenges. While the afternoon session is more focused, is dedicated to policymakers. So the focus will be more on regulation and policy and strategies for driving European agriculture towards sustainability and innovation. So I really thank everybody, especially speakers. Thank you for your participation and your contribution. And now let's have a look to the program for today. Maybe Bruno, if you can share the screen with the program. Here we are. So first, we will start with Professor Bruno Mezzetti, who's the chair of the iPlanta Cost Action. Bruno will present RNAi technology, giving an overview of its main application. Then 2 interventions on RNAi real existing application. First, Elena Baraldi from the University of Bologna will present the activity of an Horizon 2020 Prima project on the development of RNAi-based solution for strawberry production. And then Nick Vangelube, giving his talk about the market challenges of new RNAi-based plant varieties. And the final intervention will be by Alessandro Dal Piazza, the director of the Italian Association of Apple Producers, who will provide some insights on the producer's perspective and the challenges of the recent EU policy for sustainable agriculture. And at the end, we will watch together a new video on RNAi, a recently developed information tool that is released today for the very first time. And then the roundtable with 3 main interventions: Mirko Montefiori from New Plants, Anna Aldrighetti Sant'Orsola, and Peter Mellis, PCF Research Center, Oaksgraten. and then open discussion. So the program is very rich. I think we can start. If you have again any question for the speakers, please put it in the chat box. It will be possible to ask questions to each speakers at the end of each presentation, but at the end we will also have a final discussion to address more general issues. I ask all the speakers to stay within the time limit because the program is very rich. And one last important thing, with the same intent to better understand the context and the potential development of RNAi-based solution, we developed a short survey to collect data about the stakeholders' perception. So at the end of the webinar, or at least tomorrow, you will receive an email with the link to the survey. I really ask your collaboration in compiling and filling in the questionnaire, and possibly in spreading the link with your own personal networks. And be sure that all the response will remain confidential and anonymous. And I thank you in advance, all of you, for this collaboration. So I think we can start with the first speaker. Bruno Mezzetti, the chair of the iPlanta Cost Action. The title of his presentation is RNAi for Greener Food Product. So Bruno, the floor is yours.
Good morning. I think you are hearing me now. Well, thanks, Vera, again, for introducing this webinar. And well, my introduction is also really generally related to the iPlanta program. So it's for new people that is joining the webinar today. Is a consortium supported by the COST program, Horizon 2020 COST program. It's joining scientists from 34 European country, and started in 2016, and we are going to the end. So it's something that is in some way ending. We actually had almost 1 year in addition because of the COVID pandemic. The activity of today is related to working group 4, RNAi social and economic impact. And it's one of the 5 working groups that were operating in the last 4 years, and all this technology, and specifically related to the technology of gene silencing. So RNAs, that is a natural mechanism that is offering a really interesting opportunity because of the possibility of exchanging message between plant and organism, the fact that this small molecule can move inside the plant, and finally also the new things that are related to the fact that we can apply this type of product directly with the spray, so with the topical application. So practically is offering 2 opportunities: direct delivery in plants, so implant a stable expression of new RNA sequences, silencing sequencing, or for preparation of new formulates to be applied with direct spray. The technology is well known since several years. Actually, this was Melling and Fire having a Nobel Prize in 2006, but in 2010 we already have some interesting application of the technology in controlling fruit ripening and save fruit loss, maintaining, so increasing the shelf life of the fruit. And again, another example that is at commercial level is the Arctic apple, saving the oxidation of the fruit, so again improving shelf life, or metabolism related to the metabolic pathway on some nutritional compounds like glutens or allergens and so on, that are considered really interesting. And most of the action, however, was focusing on this new knowledge that has been developed by studying RNA technology that is offering a new opportunity. The RNA technology is offering in controlling the interaction between plant and pathogen disease and in both fungi and the virus, but also pests, so insect, so controlling fungal diseases and virus disease and pest diseases. And this, again, many results have been demonstrated already. So the possibility to have resistance to nematodes, resistance to insect, the viral resistance with the cultivated varieties, that is a rainbow papaya, that is for us an example, a good example for the application of this technology on fruit. And the long story related to the plum pot virus resistance in plum, so the Sharka virus disease that is very impactful. Today we will have more information about this. And well, we have been discussing the possibility to extend this technology also to protect other, to control other emergency, like can be in Italy and in Mediterranean country, diseases related to Xylella fastidiosa. This is still is a limitation because this system is not really so efficient on bacteria, but it can be a development, a new development. But in any case, an opportunity could be to use the technology to control the vectors. That it's in some way also important to reduce the impact of this disease. For working for fruit crop or grafted propagated vegetable propagated plant or grafted plant, we see also an interesting opportunity to have the rootstock as a source of production of RNA that can move in the plant and have and transfer in the fruit, in the canopy, the resistance to some diseases. Is something that many groups are now focusing because they see a very interesting opportunity on this, in particular because of the fact that you have stable iPlanta production of RNA. We will see that this is through a genetically modified approach, but it will remain only in the rootstock, only in the root. So the only part genetically modified will remain the root and the rootstock, while the other part will not be genetically modified. But just the small molecular moving, this natural molecular moving from the root to the canopy, providing the resistance. So we think that it will be more easy for public acceptance. Everybody knows that we are having big concern about pesticides or the need of reducing pesticide. We will discuss a lot today, also from indication from the different company. And I say that realize that the second application, the second opportunity offered by double-stranded RNA can help really a lot probably to reach this goal. So the fact that we need to maybe we can develop new product based on RNA technology, create a new formulates with specific with RNA sequence that can have specific effect in controlling diseases without having unintended effect to any other microorganism. And this at the moment is clear that can be applied to protect insects, to control insect diseases, to control fungi diseases, still limit its application to bacterial diseases. But then we can also have other applications, for example, controlling fruit ripening or other aspects of fruit development. In terms of diseases, well, always the most presented study is a advanced study, and actually again Elena Baraldi will give some more detail about this, the possibility to control botrytis infection with this application in both approaches, so a stable expression in plant, so Higgs, and topical application with new formulate of RNA based product for controlling botrytis infection. And this first paper was demonstrating the efficiency in tomato, strawberry, and grape. The other important case study is Fusarium in cereal, and the group in Gießen are really active on this, but the other group in the world, and people in Ghent that were demonstrating also the capacity to control Drosophila and Drosophila Suzuki and other emerging diseases, pests we will see impact with a high impact in different cultivation. Clearly, this approach, it's quite large approach. You need to develop the technology, the sequence, but then also to develop the new product and formulation. So industry working on this are focused now to putting a lot of effort, not only to design and identify the specific sequence, but also to create the formulate that can be more efficient in increasing the stability of the RNA sequence and the efficacy of the RNA sequence in the absorption and activity in controlling the disease. In fact, this the delivery is one of the main factors, one of the main aspects that you have to consider in developing this technology in this new type of product that with a high efficiency and also sustainability, so reducing cost that can be accessible to the consumer. Just to go to the end, this is really a general first introduction in terms of risk assessment is something that we will consider this also. It has been really impactful in the discussion we had in the last 4 years. We see the fact that by applying RNA technology, the risk assessment can be at least focused on some aspect. Clearly, the benefit is the fact that we are not expressing or applying new kind of protein or other, let's say, unknown product. But what we develop, what we focus here is on the development of clearly new specific RNA sequence that can be well defined, can be tested also preliminary with bioinformatics study in order to see if there are some kind of unintended and of risk of unintended effect or off-target interaction with the genome expression of the different target organism. And so this should simplify because we should not consider the fact to analyze the effect of the new substance because we are not introducing, we are not spraying new substance, but we are just spraying new small molecular, natural molecular. So all this discussion about the risk assessment will be also considered today in the afternoon with the presentation from EFSA. So we see, and we are here today to discuss this technology because we see some benefit as a potential new natural molecular offering new opportunity for plant improvement and protection. We are not producing new protein, new enzyme is only nucleotide, very short, specific, identified nucleotide. And if you have the endogenous applications, the approach can be durable. So you have a stable resistance. You can induce a stable resistance. You can downregulate genes and so manage the different type of metabolism depending to your goal, and this RNA we have seen that it's also systemic, so you can control different metabolism or different type of development of the plant or diseases that are affecting the plant at different level by, for example, using even only the root stock. And the alternative technology that we are now looking for with much higher impact is the fact that double-strand RNA can be used also for topical application. So to develop new product, RNA-based product that can protect. So two solutions. The first one is a stable resistant plant. The second one, new product, RNA-based product that can be used to replace traditional pesticide. Yeah, the last one, so the spray just to be clear, is not, we are not modifying the plant. Why? If we have a stable expression of RNA in some way, we are modifying the plant, but as saying in a different way, if we are using the rootstock in some way, the plant is not a GMO. So I give this last slide before thanking you all as a point of discussion on the application of this technology. This is a network that we reached in few years with the traditional system before COVID pandemic situation, and we were already more than 200 researchers involved with the webinars. Now we are almost near to 900 researchers involved that were following all these things. So in some way at this point the webinar were at least of interest because we had the possibility to even enlarge our network and contact more people and so on. So thank you again for your presentation, and I hope this was helpful for introduction to today's webinar.
Thank you, Bruno. I don't see any question in the chat. If some of you wants to ask something. Otherwise, we can move to the next presenter. Okay. So the next speaker is Elena Baraldi from the University of Bologna. Hello again, Elena.
Hello. Hello.
And the title.
Good morning.
Good morning. And the title of her presentation is Horizon 2020 Prima Medberry Project, RNAi for Sustainable Strawberry Production. Elena, the floor is yours.
I am preparing the slides. Okay, do you see them? Yeah. Do you see them? Okay. Okay, thank you, Vera and Bruno, for this invitation. I'm here today to briefly present you a project entitled Medberry, Developing New Strategies to Protect the Strawberry Crop in the Mediterranean Country, as an example of a project that I think can help to promote RNAi-based strategies, especially for crop protection. In this case, we focus on strawberry crop and on fungal diseases. Medberry is a Prima project. We will hear about Prima project this afternoon, so I think also that some of you can be familiar with this. Prima stands for Partnership for Research and Innovation in Mediterranean Area, and is a project under the umbrella of Horizon EU. So it's a European project specifically dedicated to Mediterranean countries. Okay. So Medberry is financed, was financed in 2018, the first call of Prima. The call number 2 is Sustainable Farming Systems under Mediterranean Environmental Constraints. The topic 122, Preventing and Controlling Emergence of Animal and Plant Diseases. I don't think the animation is working, but okay, anyway. Medberry involves 5 countries. So Italy, France, Spain, Morocco, and Turkey. Of course, they are in the Mediterranean basin. And in Italy, we have 3 universities: the University of Bologna, which is my university, University of Milan, with a group of Dario, Vrisio, and Vera, Polytechnic University of Marche, with the group of Bruno. In France, we have the INRA Institute and a company, a strawberry company called Invenio. In Spain, we have the University of Cordoba and the company Viveros California for a strawberry company. And then in Morocco, the Institute of Agronomy and Veterinary, Hassan II. And in Turkey, the University of Kukurova. I don't understand why the animation is not working, but I don't know how to solve this problem, but okay, I will try to go on anyway. Okay, this is a 3 years project, so it started the 1st of September 2019, and we are actually exactly in the middle now of the project, and so we are halfway. The project general aim is to provide alternative and sustainable solutions to reduce drastically the use of toxic pesticide for strawberry used in strawberry disease management. In particular, we are tackling 3 fungal diseases, gray mold caused by Botrytis cinerea, powder mildew caused by Podosphaera fanis, and anthracnose caused by Coletotricum species. These are 3 major fungal diseases causing huge economic losses when the crop is not managed with a number of agrochemicals. So we want to reach this aim through 4 experimental VP, which is the VP1, valorizing local germ plasma through breeding activities, VP2, developing intragenic strawberry, and then a VP3 that is a very interesting VP for this cost action, since it involves the development of RNAi-based strategies as protection tools. And then a VP4 that analyze the socio-economic sustainability of this innovation that we want to propose with this project. And of course, VP number 5 that is dedicated to communication and dissemination activities. So I go quickly through this VP. We have VP1 that I said is mainly on breeding activity. Here we are exploiting and valorizing already existing genetic material present in the countries that are participating to the Medberry consortium. And here we have already evaluated segregating populations and obtained interesting breeding lines and resistant genotypes that are currently under evaluation in the field in different countries of the Mediterranean basin. Then the VP2, as I said, is the development of intragenic strawberry, intragenic resistant strawberry. So what is it? It's a strawberry plant in which we make genetic manipulation of its own gene. So they are genes belonging to the same species or closely related species. We do this using, instead of kanamycin resistant gene, we use a gene that consists of a MIB reporter gene that confer a red color to the plants, so is useful to select the transformants. And these plants are programmed to express defense genes that will work against botrytis and Coletotricum in an inducible manner during the fruit development. So they will protect, as we expect, they will protect the fruits from gray mold and anthracnose. And in this case, this picture comes from the University of Cordoba and also University of Marche from Bruno's lab, where we already obtained some of these red colored plants that are promising for intragenic achievement. The Whoopie tree, which is the one interesting here, of course, is the one where we exploit RNAi interference to achieve the two approaches that Bruno clearly presented in a previous presentation, that are the endogenous approach, the host-induced gene silencing, and the exogenous approach, the spray-induced gene silencing. So we want to achieve this in a strawberry. Here in the left, of course, you see the RNAi molecular mechanism. I'm not going into the detail of this mechanism because we hear about this already many times. But I will tell you more about this WUPI. So this WUPI has 3 tasks. The first task is the preparation of the genetic material necessary for eggs and SIGs. The second task is the development of eggs, and the third is the development of SIGs. So in the first task, what we did is an in silico analysis that allowed the identification of the most interesting and efficient targets that are genes necessary, important for the life of fungal pathogens. So what we are exploiting here is that cross-kingdom RNA interference that Bruno presented before. And then we went on by preparing a construct for the development of eggs plant, so strawberry plant genetically transformed for expressing this hairpin construct tackling the fungal genes. And then for producing for the SIGs approach, the exogenous approach, producing double-strand RNA, using both heterologous system like coli and tobacco, but also in vitro transcription and external service. So in this case, I'm not going to present all what we have program and what are these activities, but only the most advanced activities. Here we actually, we made 3 constructs for 3 types of strawberry plants resistant for the 3 different diseases. And in all the 3 cases, we went to agrobacterium transformation, and we are now under in vitro regeneration. For some of these constructs, specifically the one against botrytis, we already reached, we already selected the transformant that express the hairpin construct necessary to silence the fungal gene. And we already obtained plants that are now under acclimatation in the greenhouse, and they are going to be tested for susceptibility again, for botrytis very soon. These are pictures coming from Bruno's lab. Okay, for a SIGs approach, we have developed 6 constructs for producing the 6 double-strand RNA against botrytis. one for Podosfera, the causal agent of powdery mildew, and 3 for Colletotricum. Only one for Podosfera. The reason is that the genome here of this fungal pathogen is not sequenced, so we had some trouble to actually amplify the key genes, the target genes, with the generate primers. But at the end, we managed to get one. So what I present here is the results from Bruno's lab that are concerning the construct against the botrytis, which is the one at the most advanced stage. And actually here we reach a quite good TRL level, I would say around 6 or 7, because these plants, these strawberry plants that are sprayed with the construct against botrytis are in the greenhouse, so it's a relevant environment for strawberry production. And what we are doing here is to test the best, to find out the best concentration for using this molecule, the double-strand RNA, for achieving protection against botrytis in a very similar way, or even a better way than the standard fungicide. So both in terms of disease severity and disease control value. Here below you can see the picture of the plants in the greenhouse. We have the negative control sprayed with water with a lot of damage in the fruits, and then the different concentration of the double-strand RNA treatment going from 10 to 50 nanogram per microliter, and then the positive control, which consists of fungicide treatment. And you see that is quite comparable with the last dsRNA concentration of 50 nanogram per microliter. Okay, so very quickly, we also have this, of before, which is a very important work package, because here we have to analyze the sustainability of our innovation. Here we have 3 tasks. The first one is already accomplished, and is the analysis of the economic background and cost benefit of the strawberry market worldwide. So it consists of the base where we can place our innovation. And then we are going to go on with the task number 2 for the economic sustainability of the solution, particularly for RNAe, which is quite new, and for the consumer acceptance, so the social acceptance of this solution, which is compulsory for driving these strategies based on RNA into the market and into society. So in conclusion, we are halfway of this project, Medberry, that I have the pleasure to coordinate, and already our data are promising in terms of showing that the RNA interference-based strategy are a very good candidate to substitute toxic pesticides for controlling important diseases in strawberry. Of course, we need now to analyze the economic impact and the social impact of this solution, and also risk assessment studies will be necessary for the application of this innovation in the field. I want to thank all the people participating to this project, and you, of course, for your attention. Thank you. If you have any question, I am happy to answer.
Thank you, Elena, for this very interesting presentation. There's one question in the chat. Wait. From Martin Sommers. The question is, in general, is exogenous application as effective as transgenic approaches, or what is the trade-off?
Okay, there are advantages and disadvantages. Of course, transgenic application, okay, we still have to collect many data about it, because what I presented here are the last experiment from Bruno's lab, and so they are very preliminary. And but in general, the transgenic Higgs approach is of course more time consuming and more energy consuming, because and depending on the crop, not all the crop are of course prone to transformation. Some are recalcitrant, we know, and generally they need quite a long time to be obtained. But of course, then we will have the advantage of have a stable and long-lasting, so permanent protection in the plants. Whereas for the exogenous application, we have to play with concentration, and is going to be also the economic impact is going to be for sure higher at the end, because we need probably more application of this product, and many protocols are still to be set up. However, our preliminary results show that some of these molecules have both preventive and therapeutic potential, so they can protect the plants before and after the pathogen attack. And I think yeah, we have to go on with the study to be able to respond to this question in a very precise way. What I'm saying here are very general consideration, because at the moment this is what I can do.
Thank you, Elena. There's another question from the chat from Aileen. For your 6 experiments, have you added a detergent adjuvant to the dsRNA, or was it just mixed with water?
Okay, I think this is yes, they is experiment from Bruno's lab. Yeah, I maybe Bruno can answer.
In this data, we used water only, but yeah, we are running experiment also with some kind of solvent. At the moment, there are preliminary data, and we cannot provide any information at the moment. But that results were with water.
Thank you, Bruno. Yeah, exactly. Okay. Yeah, Silvet, exactly.
Yeah, or answer.
Okay. Any other questions from the audience? Okay. So I think we can move on with the program with the next presenter, Nick van Geluwe. Sorry for my pronunciation. And the title of his presentation is Regulatory and Market Challenges for RNAi Products in the EU. Thank you, Nick.
Good morning, everyone. So I'm Nick van Geluwe, and I'm a collaborator of the PRESCA Association. That stands for the Public Research Specialty Crops Approval Association. And today I would like to address to you some mainly, let's say, regulatory challenges related to RNA products in Europe, with a particular test case, and that is the honey suite, an RNAi-based PPV-resistant plant. So in general, we have, let's say, 2 main applications. We have the RNAi-based resistant plants, and they are obtained with DNA recombinant technology, and so they are regulated as GMOs. However, these GM plants, they differ most of the other GM plants because of the stable expression of double-stranded RNA molecules, and no other novel or exogenous proteins as active molecules. So in addition, the expressed double-stranded RNA molecules, they are highly mobile in the plant vascular system. And so in some cases, vegetative propagation of horticultural crops by grafting only the transgenic rootstock that can also contribute to confer resistance to the entire plant. So this type of double-stranded RNA application is of particular interest to the seed and the nursery industries that are actually demanding good, new resistant, high-quality varieties. The second option, RNAi-based plant protection products, well, they can also be directly applied using current agricultural practices such as spraying. At the moment, only a few products are currently available. However, many promising RNAi-based molecules are under development, as just illustrated before. And what is an important aspect here is the regulatory challenge that we see for these RNA-based applications. So for example, for the RNAi-based plant protection products, there is a lack of categorization and regulatory experience in Europe. So the European Food Safety Authority, EFSA, they only have recently commissioned a review of the evidence of hazards related to the use of RNAi GM plants. And although EFSA has not focused on exogenously applied double-strand RNA, there is an overlap in safety data from RNAi-based resistant plants that could serve as a valuable input for the review of the safety hazards also applicable for exogenously delivered double-strand RNA. And that's where honey sweet plum comes in case. So actually, since 1990, the EU rules for GMOs, they have changed several times, with the most recent regulation on risk assessment transparency. However, since 2012, there have been no submissions for cultivation. So the honey sweet dossier can be a good test case because of several novelties. So first, the product is developed by the public research sector. Second, it's a perennial tree instead of an annual crop plant. Third, we have here the concept of RNAi interference that is being used as a tool. And then also HoneySweet provides an example of cultivation and consumption of an unprocessed product. So the aim is that the experience with the regulatory process will be shared with public sector research institutes. There are different paths for the submission of the HoneySweet dossier. So for placing on the market of GM crops in the EU, there are various paths as depicted here. In the first part, you go for a food feed regulation, and the second case for cultivation. And then you can also do it separately in two steps. So for food feed and cultivation. And so what we are aiming for with our association is to follow path 3B. So first, there will be submission of the cultivation dossier by the competent authorities from the Netherlands, followed by a submission of a food feed dossier by the Czech Republic competent authorities. And so this will be submitted by the Presca Association. So it's an international non-profit association, which stands for, as mentioned earlier, a public research specialty crops approval association. And what is the purpose of this association? To collaborate on facilitating the availability of genetically modified crops that are developed by or with the contribution of public research institutes. And we are involved in all kinds of activities. So now, as a test case related to regulatory challenges, I would like to show you the preparation of the dossier for cultivation in the European Union. So this is the Dutch application form for the GMO dossier that is available on the website of the competent authority. So as you can see, this is only 6 pages with questions. However, what is being asked of us is a tremendous amount of work. In general, there are 4 main aspects related to an application form. first aspect is general information. Then what is the nature of the genetically modified higher plant that is contained in the product? And then in C is being asked information about potential previous releases, think about field trials, etc. And then in D, information relating to a monitoring plan. So in this presentation, I will just shortly touch the different aspects that are required if you want to bring an RNA-based application to the market. So for example, general information that is also about, let's say, market possibilities. So what is, for example, now being produced? And so in the case of plum, Romania produces almost half of the total production volume in the European Union, followed by Italy, France, Spain, and Poland. Then in the next case, we provide information about the nature of the genetically modified higher plant. And here we have to provide really a lot of different information. So we have to provide information related to the plant that will receive the genetic modification, then information related to the genetic modification, let's say the construct, and then information relating to genetic modification introduced in the recipient, so in the genetically modified higher plant. And then as a final aspect, we have to provide information on the potential environmental impact from the release of the genetically modified higher plant in the environment. So this relates to the risk assessment. So what is actually known about this honey sweet? So just to give you a short intel on this, the cultivar is bluebird, and so this is a hexaploid species and is self-sterile. Then furthermore, in this dossier, you have to explain the sexual compatibility between cultivated and wild species. And then furthermore, you provide information about survivability, dissemination, and interaction with the environment. Is this, for example, important for pollinators, etc. And then you also describe the phenotypic and the genotypic traits. This is still, let's say, a lot in the comfort zone of many academic research institutes. Then we have to provide information relating to the genetic modification. So here you have an overview of the DNA construct. So it's a classic tDNA with different expression cassettes. So the plum has been transformed using agrobacterium-mediated transformation. And so it confers a gene expressing the code protein of this plumpox virus. And what has happened to make this plant develop, there was a co-cultivation with hypocotyl slices of plum, and then a new plant was regenerated. So here you provide all necessary information related to the tDNA. As you can see, there is a GUS construct. There is also a kinemycin resistance selection marker, etc. Then in the next step, you have to provide information relating to the genetically modified plant. And as people who have done plant transformation before with agrobacterium, we are aware that this can be pretty complex, from one insertion to multiple insertions, etc. So in the plant that has been selected, it was identified that there are actually 2 insertions. And in the first insertion, there are 5 partial copies and 2 complete copies of the tDNA construct as depicted here. In the second insertion site, there are 2 partial copies who are in an inverted position, which results in a hairpin structure. And so it is proven that it is this hairpin structure that results in the RNAi based application of this honey sweet plum. Furthermore, you have to provide information about if these insertions, they do not impact other genes present in the plum genome, etc. But what I want to show you here is also a very important aspect for this dossier, is to show the case that there can be no coat protein of this virus detected in the honey sweet or in this cultivar, as you can see, compared to other samples. So in summary, the honey sweet plum does not express the coat protein at detectable levels, which eliminates the concern of protein exposure to potential non-target organisms. And I think this is a strength compared to other GM plants that are, let's say, applied for evaluation. And then furthermore, you have to provide a lot of information related to the genetically modified plant, which is, for example, the fact, how does it differ from the recipient plant, the potential transfer of genetic material to other organisms, is there a potential? And also, for example, the case if this would happen to microorganisms. And then the final thing is providing information on the potential environmental impact from the release. And so this is nothing else than risk assessment that is being done. So a question that is being asked there is, for example, what is the likelihood of this plant becoming more persistent than the recipient plants? Yeah, or could it be more invasive in natural habitats? Is there any selective advantage or disadvantage conferred to that? So this is risk assessment that needs to be done in a step-based approach, and where it is important, where we have people involved in the Presca Association that help us with that, because this is more out of the, let's say, classical academic comfort zone. And then in the next aspect, you have to provide information relating to previous releases. So what is relevant here is that there have been field tests conducted in the US since 1995, and that there is an unregulated cultivation possible in 2007. And furthermore, we have a lot of field testing that was done in the European Union, such as Czech Republic, Romania, Poland, and Spain. And so in summary, no immediate or delayed adverse effects as a result of the direct and indirect interaction of the genetically modified plum with the environment has been identified. And then in a final step, you have to provide information relating to a monitoring plan. So in general, a general surveillance plan is mandatory, because imagine that something would go wrong with your plum. This might result in the occurrence of unforeseen adverse effects, and you need to be able to, to follow up on that. And then furthermore, it's possible that a case-specific monitoring is required. If you would have identified a particular problem, such as, for example, there might be problem with pollinators, then it can be the case that you are asked to provide a case-specific monitoring plan to follow up on this. However, in our case, we think this is not necessary. Furthermore, related to regulatory challenges, an important aspect is that EFSA has now come up with a new transparency regulation where you need to fulfill to when you submit a dossier. And so I provide you here with a link for more information. So I would like to thank Presca for the nice collaboration. And in case you would have any questions here, I provide you with my contact details. Thank you for your time.
Thank you, Nick. First of all, I ask all the participants to mute your microphone because someone has the microphone open. Thank you. Are there any questions for Nick? I cannot see from the chat any question. Okay, so thank you, Nick. Thank you so much for this very nice presentation. The next speaker is Alessandro Dal Piazza, the director of the Italian Association of Apple Producer. And the title of his presentation is Green Deal or Green Dream. So thank you, Alessandro, and the floor is yours.
Thank you very much. I try to share my screen. You hearing me? Yes?
Yes, we can.
Okay, thank you. Here it is. Yes, thank you for your kind invitation. It's a pleasure to be here with you today, as we are, what to say, every day dealing with the pesticides problem, large area of problems, and not only from the technical point of view, but even pressed by the public opinion that are increasingly concerned about the reflections of pesticides use on the citizens, consumers, and so on. So we are really, really deeply involved in these topics. The title, just to avoid misinterpretation, we are not against the Green Deal or the Farm to Fork, but we are concerned about the way authorities, subjects involved in this process are right now proceeding. So we feel that there are something that should be corrected in order to reach the goal, not to oppose the goal. What I would present you is a very brief example of the Italian apple producers. It isn't a solution of anything, I would say. It's something that I hope and suppose could be helpful for you to understand what's behind the scene on the from the production side. So it's every time good for me and for us to bring the voice of the producers as well. Let me start very briefly introducing who we are. Assomela is an association participated by 13 producers organizations that are a big consortium. And through their base, we represent around the 75, 80% of the global whole apple Italian production. That corresponds to the 20% of the Europeans one, more or less, depending on the year, depending on the will of the god, I would say, no frost and so on. Among our activities, there's the one highlighted in red, you can see there, to coordinate activities of the members in research, innovation, and technical assistance sector. Inside this precise task, there's the field of the sustainability and the pesticides as well, finding or trying to find solutions progressively better in order to implement the best way we can the technical procedures in the fields are of a general and common interest, and so are based on Assomela. We are dealing with carbon footprint as well, management of water, and so on. Many of the sides of the sustainability. Where we are. Assomela means apple production, so they are concentrated in north of Italy, most of all in Trentino and South Tyrol, where through the action on the role of the three bigger producers organization, around the 70% of the Italian apples are brought to the market, half of them at national level, the rest exported worldwide. So we can assess around 90 countries worldwide. And I come to the topic of today. You all know probably the main targets of the Farm to Fork and the Green Deal. I don't mention. I simply remind the first one because it is precisely what we are speaking about today. To reduce by the 50% the use and risk of chemical pesticides. Risk is important to mention because we can't judge and evaluate the progress only in terms of volume, kilos, and so on, but also through the related risk that are connected to the use of pesticides. Why I'm saying that? general remarks, and I take 2-3 slides from one presentation of the European Commission last October, just to frame the concepts I'm speaking about. Important in this process should be to assess constantly where we are and where we go. But we feel that the impact assessment is still lacking in some way in the action of European authorities. And just to come back to the 50% of reduction, we really don't understand who proposed the number. Why not 45 or why not 60, for example? And so we suppose that there's behind a very in-depth study about the feasibility of this goal. But as far as I know, there's something moving, and this supposed lack should be recovered in the forthcoming weeks or months. It is a policy, it is a strategy, the Green Deal and the Farm to Fork. And as you can see in the first line, targets are today aspirational and not binding. But as we can see further in the presentation, many authorities are acting as if the thresholds and the targets were already binding or were already in the law, and not only a strategy and a philosophy, let me say, in some way. In this slide, I bring your attention on the second box and the others, where we can read a revision of the sustainable use of pesticides. The pesticides directive is among the first priority. We are right now dealing with an open consultation that will end at the beginning of April. But important is to mention the enhancement of integrated pest management. It's a goal already among the activities of the European authorities, and for us it's really strategic because all our production is covered by this technical integrated pest management protocol. The state of play of the IPM related to the phytosanitary means, because our IPM is larger and includes other aspects that are important for the daily work of the producers, and it's limited to the scab in this case, not other pests. And so it's really a limited machinery I'm proposing to you. At this moment, we have 3 active substances under discussion. Or better, the first one, mancozeb, hasn't been renewed last year. We will use this year up to the end of 21, but then it will be not more available for the producers. Captain, I don't know if you know, but really strategic for the IPM. There's an ongoing discussion on renewal with possible limitation to be used in greenhouse only. It would mean that in the open field will not be usable, and with very severe consequences for the IPM protocols. The second one, on the third, is dithianon. The authorization will expire in 23, and so there's a bit of time to discuss this point. But just at the starting of the discussion, similar limitations are foreseen as like Captain, and introduced to be used only in greenhouse in the future. And so the consequence would be very similar. What does it mean? First of all, are mainly preventing active substances, not very easily to be substituted. Today on the market, in the case of apples, I mean, there aren't very effective new active substances already available. And they are also, I mean, Captan and Dithion, very well known, historically used, no risky for the producers, but also how we will see just after this slide, very secure, very safe from many points of view, I would say. Captan renewal process, there's an estimation for the economy linked to the Captan use or not use. Currently, the apple production is spread on 500,000 hectares, more or less, at European level. And the marketable production is not 14 million tons, but around 11, 12, depends on the year. Even in this case, the rest is directly addressed to the processing sectors. Captan is used to the 20% of the producing area. That's why it's very effective against scab and gliosporium and some pests that are that can be dangerous for the storage of the apples, and it's used very often 2 times, 3, 4 times more or less just before the harvest to control and to prevent those pests. Period of use I've just mentioned, the number of treatments, and the pest Venturia inequalis, Neonectria dittissima, and the Nectria galligena as well, and Gliosporium, very important. Why I've said just before that the use is safe. As you can see here, those are the analysis made on Captan to control the level of the residues. On the left box, you see that in the 85% of the cases, Captan is detected. But on the right box, you see that in the 99% of the cases, the MRL are lower than the 30% of the maximum allowed residue, by law, I mean. And in our opinion, it means that all the provisions and the limitations, the indications given to the growers through the IPM protocols are effective, are respected and effective. It means that using with intelligence this active substance, it's important to give to the consumers the highest level of security, safety we can offer to all the community today. The second one is the water quality impact. I'm mentioning because one of the problems highlighted by the European services right now to support the proposed limitations should relate to the environmental impact. Water is a part of the environment, and so it's important to go a little bit in depth in this part of the dossier. The previous data are collected through the producers' organizations, the most important at Italian level. The water quality impact comes from the work of the ISPRA. ISPRA is an institute active, national and public, active in the research and the mitigation of the environmental problems. And so it's an official source. In this case, as you can see here, for the superficial water, limiting my comment to the 18, starting from the 1,500 samples made, only in 8 cases the Captan has been detected, and only in 1 case the limit was over the maximum threshold for the residue. And that's the scenery that we can see for the 17 or so, but going and reading other of reports of the ISPRA, the scenery is more or less the same. we comment the results as follow. Captain residue findings are low, and the low is the level of the residue. It means, once again, that the provisions given in the IPM protocols and the use of technology solutions are able to control and to mitigate the drift effect, and as a final result, having controlled the drift, even to reduce so strongly the interference of captan in the water. And so it means that this active substance deserves to be better analyzed before to say we take out from the market or we put important limitations. I've heard with pleasure that the economic side of your work is taken in consideration, and so I propose the same method, taking some results from work and assessment made by a private company. I don't mention the company, but simply to show you how important it is, or how big and severe can be the consequences in taking out one molecule, only one in this case. As you can see here, that has been made in several countries, but I draw your attention on Italy here. Simply not having available the availability of captan, we could estimate a loss around 20% in the yield of apples for the fresh market. And passing to the economic estimation, it would mean for Italy around 37% of economic loss. I don't know if the counts are agreeable, shareable. In my opinion, maybe there are an exaggeration, because in some way we can mitigate with other ways this lack of captan. But in any case, independently from being 10%, 12, 15, or whatever, it's an important loss for the producing sector. And so this way of approaching the estimation and every policy in Europe should be accompanied by a very in-depth and detailed impact assessment before to take the final decisions. What would it mean, this kind of process? About the IPM, I would say it will be a stake, because captan and DTN on the second one are the pillars today of the integrated producing methods. Not having them, probably will use more systemic active substances that implies in the short future an increased level of resistance, tolerance against the pests, and the necessity to multiply the use of other pesticides. And so it's an open door to the opposite goals, instead of having a reducing the use of pesticides. But most of all, it will be the farm viability at a stake. As you saw, the economic loss can be high, but think to the necessity to spend more, to use more active substances, to control many other processes, and it's for sure that the farm viability can strongly suffer of this process. Direct value, because it is the global turnover made by the associations, that's the sum of every single turnover of every single farmer. The indirect value, because not having on the territory the presence of thousands of growers, I forgot to mention that Assomela represents around 15,000 small growers. And so can come from this effect, loss in terms of occupation, for example, and working places, and social impact can be really high. Territories degradation, not being on the territory, every single farm to maintain and to see what's happening in their fields, it's for sure that in a short term, in the short run, negative effect on the territory will be present. And so even this point has to be taken in account in the general policies. I come to what? How to prevent or to correct this direction. First of all, we think that advanced technologies are really needed. Some of them are already on the market, like anti-drift nozzle that can control the effect in terms of environment, overhead spraying tools that are emerging, are coming, are already available, but not yet very largely implemented, or decision support system, precision farming, and so on. We have heard several times the concepts. All in all, technologies are needed. Appropriate mitigation measures that can contribute, buffer zones or timing in the morning where some intervention can be made instead of others, and so on. And the producers are completely open to adopt them. Research and innovation, biopesticides, I've heard something in the previous presentations. RNA, for me, is like to come back some years, because at the very beginning of my professional career, I was in the fields with pollution and with cross-pollination to find out resistance, or try to introduce resistance in the apple sector. And so if those solutions will be available, we will be for sure more than happy to implement. Or smart genomic technologies that are today accepted and wished by the producers. We know that this field is quite complex, and not all at a political level are in favor of this technology. We think that posing very ambitious goals like reducing the use of pesticide, but not at the same time offering to the global community other technological advanced methods, it's a little bit, I don't say that, is contradictory. Right pace of the process, because taking out old substances without having new technologies or new products already available at the same level of efficiency, it's like to say we are going in the wrong direction instead of the right. And the impact assessment methods inclusive of economical implication for farmers. I attended several meetings in the sustainability area and so on, but I would say that very rarely this component of sustainability concept is highlighted. Environmental parts are in-depth analyzed, but the possible implication for the economy of every single farm are not at the same time considered. That's the way to go towards a Green Deal and not a Green Dream. That's my personal statement, but it is also my personal wish, and I would say it's the wish of all the producers. Really reach the Green Deal and not simply stay on one dream. Thank you. And sorry, I conclude saying to everyone that for some questions, maybe later on in the early afternoon, because I unfortunately have to pass to another call based on bees, like to say to you how open we are and how in-depth we are involved in the environmental and sustainability topics. Thank you once again, and sorry for passing to another meeting.
Thank you, Alessandro. Very, very interesting presentation. Thank you for stressing the importance of economic sustainability, and the global sustainability needs to include the economic aspects. Thank you so much. So we are waiting for you to open the discussion. Thank you. So Now I think it's time to, yes, if you, perfect, thank you. Now it's time to present sorry, okay. It's time to present a brand new video, which has been developed by within the iPlanta cost action, with the aim to show in a short time and in a very simple way what RNAi technology is and the benefits related to the use of this technology in agriculture. So Bruno, would you please show the video?
Yes, thank you. I share the screen. And yes, I hope that this video will be in line with the message that we just got from Alessandro Del Piaz. I now open and start the video. It's 5 minutes.
Hey, oh yes, I'm talking to you. Just a quick question. Ready? Could you imagine a world without tomatoes? Could you imagine a world without barley? Could you really imagine a world without bees? I guess your answer is no. Just like the European Commission, which has established the Farm to Fork strategy, a pillar in the Green New Deal. Among its aims, there is a progressive reduction in biodiversity loss through a 50% dismissal in agrochemical inputs, such as pesticides. The ultimate aim is to reach a more sustainable and food-secure society by 2030. Hi, Mark. This is Mark, a grower who takes to heart global sustainability policies for agriculture and food. He wants to ensure sustainable food production without using contentious pesticides. But still, plants need protection. Growers need tools to protect them. And the whole world needs food, as well as security and safety. We need a safe, efficient, and cost-effective alternative. Mark can choose between two options: stop using contentious pesticides and give in to pest infections and production loss, or stop using contentious pesticides while giving a chance to new technologies. Don't worry, Mark. Among new technologies, there's one called RNAi-based biocontrol that uses a well-known biological process in most living organisms and protects plants with a safer and more sustainable approach than contentious chemical pesticides. Did you know this? RNAi molecules can regulate gene expression by targeting specific endogenous mRNA molecules and can be designed to target genes that control the infection and disease development of pest and disease. RNAi molecules impact neither the environment because of their high and fast degradability, nor other organisms, including consumers, because of the well-controlled risk of off-target effects. Imagine you're in front of your vineyard on a rainy day. This is what you did until now. Damage the plants, making use of the heavy knapsack sprayer more than once because most fungal diseases are favored by moist conditions. This is what you can do from now on. Exploit this new technology through exogenous applications of formulated RNAi-based products, such as a spray applied only once. Or even better, to benefit from the in-plant delivery RNAi through GM crops. One technology, two opportunities. Either pest and disease protected plants or pest and disease resistant plants. And there's more. Mark isn't the only one who benefits from this, but the consumer does as well. This technology isn't dangerous for human health or the environment. The European Commission preserves biosafety thanks to the control of EFSA's specialized panel on RNAi-based plants, and plants protected by the exogenous application approach aren't considered GMO. Besides that, the two approaches offer ease of design and test, high efficacy and specificity for different pests and diseases, fast degradability, good predictability and control for potential risks. The RNAi-based technology produces new attractive products for seeds and nurseries industries interested in spreading and trading resistant and higher quality plants and products. The agribusiness sector is already investing in this technology and looking forward to developing RNAi-based resistant plants and RNAi-based products that protect plants, in both cases with high biosafety profiles for consumers and the environment. So, as you can see, aims from farm to fork strategy are achievable. Solutions to the problem of the reduction in contentious pesticide usage already exist, and they're at your fingertips. Tomatoes, barley, bees can keep on living, and us as well, but in a more sustainable and food-secure world, embracing safe, efficient, and cost-effective alternatives, like the use of RNAi technology. All it takes for a real change is to get informed, and it'll be clear that think green means understand green and look at real green solutions.
Thank you, Bruno. So the video will be available on the iPlanta website for dissemination. And I think this can be a great tool to spread information on RNAi, so feel free to use it and spread it in your personal networks. I would like to thank the authors of the video and as well as the cost officer who supported us in the making of this video. Bruno, if you want to add something.
Well, thank you, Vera. I don't know if there are comments from the people. We were trying to summarize many of the discussion we had during the 4 years of the project, together with the different groups and also with the stakeholders. In fact, we were trying to get the message that actually Alessandro Del Piaz was anticipating this vision. So it's important to find a new solution, but before to take decision, you need to really assess the new solution, to have the new solution or assess the impact of the decision that we are taking. So I think that it's really important. And we think that from the 4-year discussion that this technology can help in some way to solve some of the problems that agriculture is having in the next period. But we are open also to have some kind of comments from the people.
Okay. So let's move on to the roundtable. We will have the 3 last interventions and then open discussion, 3 questions for the speakers. So the first intervention is by Marco Montefiori from New Plant. Mirko, are you there? Okay.
Yeah. Good morning, and thank you for inviting me today, but even more important for the webinar series, which I've been able to attend as a listener or seeing and following in these days, which was very interesting for us that are on the field. I want to thank also Alessandro for the introduction that is done as a producer and the people of the industry, which has provided a very clear illustration of what is some of the concern with the new regulation and with the new strategies that we are going to, that the new Green Deal is proposing and now is drafted at the moment. Let me introduce myself or the companies that I work with. Only a few of you knew already who we are. New Plant is a small consortium, Italian consortium, which has been created now 17 years ago. The company is small, his partners are pretty big. They represent 3 large cooperative groups in Italy. Apofruit, Apoconerbo, Orzo Fresco, which you can see in total we have, if we can say, we represent large number of fruit growers in Italy for almost 30,000 hectares of fruit crop. The goal of New Plant, when it's been created, was to work in innovation to promote new cultivar development and innovation for our growers and in our territory. And when I say our territory, we are spread out across most of Italy, producing areas, and you can see we also touch different part of the fruit industry with most of the crops that we are somehow involved. Also, our core area, of course, was pear, given that we are mainly based in the Emilia-Romagna region, peach and nectarines, kiwifruit, apricot, apple, and but there are also some other which are a bit more of a niche product, which anyway we are still strong and growing, and an important part of the industry, like can be cherries or table grapes. As I said, we work on innovation and a new cultivar, and but on fruit tree mostly. And I want to just show you, for who is not used to work with fruit tree, what does it mean breeding new cultivars in on fruit tree. It's a long and tedious process and expensive process. Is in the most successful example, it will take probably around 12 to 15 years before getting your new selection into the market. And from there, starting to produce and to get it in the orchard and get commercialized. We start with several thousand seedlings, which are from these you select 1 to 5% individuals, which are propagated in small test blocks. And then after, then you already passed 10 years before you see this fruit and you can make your evaluation. Then after few years testing in different sites in pre-commercial block or orchard trials. Despite being such a long process and expensive, cultivar innovation is an important process of the industry and has been shaping what is the horticulture industry today. And we see particularly, I bring you a couple of examples, apricot industry. If you see today what we grow and what we produce is mainly, or what we plant is mainly the new cultivar that have been generated in the last 20 years. And 20 years sounds a lot, but remember, it takes about 15 years to make a new cultivar from when you cross it. So it's a couple of generations that you see of breeding that have been now taken place. And now we see that, the new generation are pretty much replacing what were the previous one. Similar, I can bring the example of kiwifruit, where it was pretty much monoculture till 20 years ago, which were only green kiwifruit Iowa, and now we see that the increase new planting and the increasing of the cultivation of this crop is mainly driven by the new cultivars, in this case, yellow fresh cultivar. So far, most of the innovation has been market driven. We were focusing for delivering new fruit which have a trait that the consumer can appreciate and distinguish and gain new space in the market or new share of the markets. But it's not only this, the only way that the breeding has been involved in these years. There has also been, of course, for other agronomical traits like pest and disease driven. We can say the kiwifruit one was a nice example on how this can be effective and important. Once we had the outbreak of the Pseudomonas syringae worldwide, which we call PSA. Here I just put a graphic of what happened in the New Zealand industry, that in a very small period of time, they completely replaced the susceptible yellow flash cultivar R16A with the new Gold 3 cultivar. And in a matter of few years, they explanted and replanted several thousand hectares. And that has been a very successful story on how you can introduce a new, more resilient cultivar to replace what was the susceptible cultivar. But it's not the normal way on how you actually can introduce resistance into or develop new cultivar. Scab resistant apples have been available into the market for several times now. It's been one of the first probably trait that has been or target of the fruit breeding program that has been developed. Took several long time since breeders are working on these traits, and now we see more cultivars appearing of this, but there's still a limited amount of cultivars that are really grown that are scab resistant. On the stone fruit, we see appearing now the first apricot that are resistant to plum pox, so to shaka. But again, there's a small number of this cultivar which are just taken and cultivated and taken into market. And we see that challenges and new target for breeding are actually appearing faster than what the breeder can respond to them. We see in the last few years, as I said, PSA is a kiwifruit, the brown stingbar, which has been a major pest which suddenly arrived in our territory. It has been estimated it costed probably more than 600 million to the fruit pipeline, which to the fruit industry and extended industry, for which we have limited tools to control it even today. It had affected many crops from stone fruit, palm fruit, kiwifruit, and so on. Drosophila Suzuki, which is a major pest for cherries and berries. Fog blight, we see coming back on fire blight infection, in particular in apple orchards. Stenphelium is now one of the major problem for the pear industry, which probably one of the consequences also of a limited number of pesticides that we can use. Plum pox viruses, as you well know, also you have been talking about it even few webinars ago. In apricot, we know that we have some resistance that we can introduce. In some other stone fruit, we are not so lucky, so it's even a bigger challenge. And climate change is, we started to appreciate some of the problems that are driven by climate change, or we can assume that is a consequence of climate change. Talk about pear, where the industry is, or part of the industry is starting to appreciate that the rootstock that we have been using for the last decades are not anymore so suitable with the new hot summer that we are experiencing and the limited water that we have in some areas. So there is a need to change and to address this and to adjust to this new situation. Or I can think about kiwifruit as well, where the recent problem of what we call Moria die-off is probably been a consequence of a different rain distribution, with excessive rain some part of the year, but this has affected the industry worldwide, nationwide in the different regions. And so what Alessandro already expressed as the ambitious target of the European Green Deal with the farm to fork to reduce chemicals, fertilizer, the possibility to apply fertilizers and increase the organic at least to 25%. Nice targets that everyone would say is could be a good opportunity. It could be a nice, nice objective, which we would agree. But the important and what is we are really concerned is that sustainable agriculture starts from sustaining its growers and its operators first, and so to make sure that we can still produce and work with this new condition, within these new regulations. And so, as I said, breeding, of course, is one of the tools that the industry is using and is implementing, but it's not always, it's not for sure a fast tool that we can implement quickly to adjust to the different situation. We have agronomical practice that we can intervene and use in different ways, and chemicals are an important part of the agronomical practice that we can use. But probably it's time to also talk about the new tools that we can implement and we will be able to use, because we need new tools, in particular if we are going to remove some of the old tools that we have. I bring you some one other example of plant pox virus, how it's affected some of the fruit industry in our area. Peach and nectarine, which was one of our main fruit or industry in our territory. In just a brief period of time, is the area invested to peach and nectar is more than half. Of course, there is not only one reason. There are commercial reasons as well and different other causes. But definitely the pressure of plant pox virus into the territory has put the grower to change their crop and to invest in other kind of industry, which having a major impact for the local economy. And so as a group, we already started to look for some of these new tools. We have a collaboration with Professor Mezzetti to look for the development of RNAi to control plant pox viruses. Of course, this is a long process. And Professor Mezzetti already discussed before of these different techniques and tools that we can have to control some of virus diseases, such as this case, and the use of transgenic GMO, rootstock for host-induced gene silencing, spray-induced gene silencing. Of course, you know these techniques better than me, but that is something that we were very, very interested to hear, but not only to hear, also to start to see what can be implemented into the industry, what can be becoming really new tools. New tools for the industry need to be efficient, cost-effective, but of course safe and sustainable for the environment and for us, for the grower, for the consumer. But with the strategy to reduce the number of tools and chemicals, we really need to have more science to develop and promote some of the new tools for the industry that we can use, and being less scary of the fact that there are biotechnologies, because I'm afraid we are already using a lot of biotechnologies, which I think that people don't understand because we use difficult words, which I'm sure that will scare people, even if I say double employed, mutagenesis, and so, which are normal tools that we use. So why this can be cannot be other new tools that we can use in our industry. Just sharing with you also one of the small concerns, well, not small, one of the other concerns that I have about the development of organic horticulture, which is an area where we are investing, and as of course is an important part of the market, sector of the market, growing market, which we are definitely interested. But 25% by 2030, I think that a lot of people should raise their eyebrows, because it can be a very ambitious target. I just look at a report in Italy of the Minister of Agriculture. Italy's 15% in 2019. Within the region of Emilia-Romagna, for the fruit industry, we're only 6% of what is today an organic sector. Growing to 25%, there are difficulties, challenges that are very ambitious from a technical point of view. For some of the industry, it will be very difficult to switch to the organic production, unless there will be the possibility to use new tools in organic production, which, why not? And the other point is the market really ready to accept and pay a higher cost for this larger volume of organic fruit or products into the market, because organic has a higher cost than a conventional fruit, because it is more costly to produce for the grower. So yeah, there are some thinking that need to be done also on it. Anyway, here is my thoughts, and thank you again for inviting me, and for the nice presentation that I've been seeing in this last meeting.
Thank you, Mirko. Are there any questions for Mirko? I don't see any questions, so I think we can go on with Anna Aldrighetti from Sant'Orsola.
Yes, here I am.
Hello, Anna.
Hello, good morning.
The floor is yours. Thank you.
Let me share my monitor. Okay, good morning, everyone. Thank you for your invitation. I'm glad to be here with you today. And yeah, today I would like to give you some information about the berry fruit production and its plant protection management. So I'm working in the Agricultural Advisory Department of the Sant'Orsola Cooperative. Sant'Orsola is a farmers cooperative specialized in berries production, marketing, and sale. And it was established in 1979 in Trentino, in the north of Italy. And it had a very remarkable expansion throughout Italy, becoming in the last years the leading company of the country. So me and my colleagues, starting from the assumption that market requires sustainability, transparency, and food safety. From the last years, one of our main goals is to ensure high quality product, for sure, with a reduced chemical residue. So with few treatments and sustainable management. It's not easy at all to ensure this kind of product. So we need to face a lot of challenges, mainly that berries are very sensitive crop. They have very aggressive pests and diseases that most of the time mainly fungi, become resistant to all the different active substances. Then algae species, for sure, such as Drosophila, already mentioned, and Aleomorphalis, they are taking over, and we do not have so much knowledge about them, and we are not able to manage properly. Then climate is changing rapidly, and we cannot forecast the normal development of the disease. Conditions are changing, and we cannot know properly the disease. Sometimes it's very confusing to forecast a proper model and a proper development of the pest, of the disease. And furthermore, since we are a very small part of the agricultural sector, there isn't so much literature and research founded for our piece of land, since we are not so interesting from an economical point of view yet, let me say. So one of our main issues in agriculture, already mentioned by Alessandro Dal Piazza and Mirko, is that in the last years we are facing with a lack of weapons in plant protection. Let me say that Europe it's intransigent about many active substances. And in the last years, it banned a lot of harmful active compounds since 2009, and now even more with the Green Deal already mentioned. Many substances were removed. I've been working in this sector just for 2 years, but in 2 years, Europe removed strategic pillars of our plant protection, such as chlorpyrifos, pinoxafen, thiacloprid, dithiocarbomates, and sometimes it's really hard for us to deal with many diseases. In these regulations, substances were evaluated whether they might be used safely with respect to human health, environment, groundwater, and non-target organisms. And a lot of companies decided not to even submit the dossier for the registration. And yeah, this regulation had also a tremendous impact also on the number of active substances developed and newly registered in the EU. Here in the graph, you can see that in the decade of the 2009, from 2000 and 2010, we had a very tremendous decrease in active substances that were newly registered. Development costs also doubled a lot. And nowadays, it now takes 11 years for the registration instead of 8 at the 2009. So it's quite complicated. On the contrary, biopesticides are increasing exponentially. Nevertheless, there are quite too many products on the market, and sometimes they are not properly regulated. Some of them are really not effective. And if they are effective, they are not even so selective towards non-target organisms. For example, parasitoids, mites, beneficial mites, and so on. And yeah, another important issue is that sustainable alternatives are not always compatible among them. For example, if we use the ozone strategy, for example, for the powdery mildew on strawberry, we will have some problems, for example, when we try to manage thrips with microorganisms, because ozone is killing practically everything. So some alternatives, some sustainable alternatives can interact negatively. Another critical point is that agriculture, it's a very low machine, in my opinion. farmers are not very prompted toward innovation sometimes, and most of the times they are quite skeptical when you suggest new alternatives. And they are used that for every problem there is a chemical solution, even if sometimes, let me say, just good agricultural practices would be enough to solve the problem, because chemical treatments are the easiest method and most of the time is cheaper and easier to use. Indeed, money and products efficiency for farmers have the priority. Another important point is that alternative products are more difficult to use because in some ways they need specific conditions, for example, microorganisms need their specific humidity, specific temperature, and sometimes farmers cannot understand these practical methods. And trust me, that once they are not satisfied by biopesticides, it's very difficult to gain their trust again. So after this speech, I can say that for sure RNA technology could be helpful for our berry production, because really we don't have so many weapons to manage pests and diseases. But yeah, I have also some questions for you that would they be under the spotlight of public concerns, mainly for our consumers? Would they be compatible with other practices, talking mainly about exogenous RNAi technologies? And would they be accepted by the supply chain, mainly at the beginning of the supply chain and farmers? Okay, so this is the end of my presentation, and if you have some questions, I'm here to answer. Thank you for your attention.
Thank you, Anna, for your very interesting presentation. So are there any questions for Anna?
There is a comment on...
Yes, I see that in the chat there's one comment. So first, a comment from Martin Sommer to clarify EU target of 25% of organic land represent an EU average. National organic action plans are to take into account the particular national starting point and the challenges related to particular organic production, for example, horticulture. So Martin Sommer, if you want to add something, please open your microphone. And
can I am Mirko Montefiore, can I
yes.
reply to Martin's comment?
Sure.
Which
yes.
I agree. I take the opportunity maybe to clarify also some of my comments on my presentation. I agree with Martin's comment that the 25% is not only for a specific horticulture. And I want to clarify that for us, it's organic is an important, as I said before, is an important part of our production and market, which we see growing and interested for the from the consumer point of view. It's only that for some of the industry, it's very difficult to produce in an organic regulation of with organic ruler today. And maybe the possibility to have these new tools could be also an opportunity for an organic sector to introduce new tools that today are not available, that will also improve or facilitate the way to grow some of the crops that today are very challenging. And what we see, there is a limited amount of organic production because some of the fruit industry is very, very challenging to be producing in a commercial, sustainable way under real organic condition. Just, but to make clear, I'm not against organic. For us, organic is something that we see a growing part. It's just that it's not very easy to do it with all of the crop that we are working with. And for sure, the fruit industry is one of the most challenging one. I take also the opportunity to reply some of the comments that I see from Andreas, that transgenic rootstock are not allowed in Italy. And this was only a research project that we started with Professor Mezzetti, but to see that the introduction of breeding and science is something that, of course, you know, don't develop in a couple of years or in a small period of time. But it's definitely the product is something that we would be interested in, and that was what pushed us to collaborate with Professor Bruno Mezzetti. But at the moment, it's only a scientific project, which of course we could be interested on implementing one day once we'll be allowed.
Another question, more general one for producers, from Jeremy Sweet. Are the fruit industries interested in testing new RNAi potential products? Mirko and Anna as well.
But I start, maybe I can say for us, we are really interested on testing the new products with the under the regulation that we have to comply with to test these products. But the needs for new tools, of course, the trends to reduce chemicals is undeniable, and we are the first one to be pleased if we are reducing the number of chemicals that we have to apply, also for the safety of the growers as well, and maybe also to contain the cost if that can be a most cost-effective way to produce. So I think that the industries are certainly interested to see and to test if there will be some of this product that will be available for testing, and depends with the regulation what is the level of testing that will be allowed, but the concept is definitely of interest.
Thank you.
Anna,
yes, I agree completely with Mirko, and we will be glad to test these kind of products. Also because we need to reduce our chemicals, our residues on our product, and just to say that I think that even if they would be properly regulated, we would be a bit concerned about what our consumers would say, because our main target is the final consumer, and we still not know how the market could be interested on these kind of RNA products. So let's see.
Thank you. Any other questions?
maybe we listen to the next speaker.
And then, okay. So the final intervention is for Peter Melis. Hello, Peter.
Thank you.
From the PCF Research Center Oxgraten. So thank you. And I leave you the floor.
First, I'll share my screen like the other ones did. Here you go. Yeah. So my name is Peter Melis. I work for the Research Center Hoogstraten in Belgium, and my field of knowledge is strawberries. And my presentation is actually very suiting, or it fits very well with the story that Anna already told you. It will be a nice addition to it. Maybe it goes a bit further, but it will be a nice addition to it. Let me first start with telling you how in Belgium we deal with pests and diseases nowadays, because we are using quite an intensive IPM strategy in our substrate cultivations. Soil cultivation is a different story, but today I will talk about the substrate cultivations and how RNAi can be a very nice addition as a tool for our growers. Opposed to legislation, the reason why our growers switched towards IPM was the loss of active of working rates of some of active ingredients, and mainly thrips was a very big problem in 2007, 8, 9, 10. Those were the key years that were making the growers switch towards IPM. Here you see some efficacy rates of different thrips products, and you see that a lot of them are hardly working. So they had to make a switch, and they made a switch towards predatory mites, and it worked really, really well. The big question of those beneficials was, in our country, we use short-day cultivars, so we use strawberry crops that are very short, only 2.5 to 4.5 months, and it's all with of course it's strawberry, so it's non-heated mostly. It's a cold, low temperature that our crop is growing in. Those are two very contradictory demands towards what a beneficial would like to see. So we had to look for solutions, and we did find them. Now growers, all of the substrate growers in Belgium against thrips are using different predatory mites and also aureus. Against spider mite, they are using different predatory mites. The most important one is Phytocetus persimilis. And against whitefly, they are using Amblydromalis pneumonicus, also a predatory mite. We are also using biological products against powdery mildew and botrytis, but they are not always that effective. Anna already told you that as well. The biological products, the biological alternatives for the chemicals that we use are not that strong. The strongest one I've put here on the slide is a Karma. It's a potassium bicarbonate that we use against powdery mildew. That is quite strong, so that's a very helpful product. But for powdery mildew, now we have a new technique, and it's growing rapidly. Multiple growers are starting to use it, and it's UVC lights. You see there a picture of a robot that is treating our crop during the nights, and it works very, very effective. So for powdery mildew, that will be a very, very good solution in the near future for all our growers. For botrytis, I will come back to that as a gap in our IPM strategy. Here are 2 slides on how our IPM strategy is a success. Here for spider mite with Phytocetus persimilis. So on the top you see the spider mites in the green bars. On the bottom you see persimilis in the blue bars. And as soon as spider mite is coming into our crop, persimilis will take over and we'll get rid of the spider mites. So this is something we'd like to see, and this is how we want to see it for every pest or disease. For thrips, it's the same story. So you see thrips coming from the top throughout the cultivation, but the predatory mites are there, and they will make sure that thrips is not causing any damage or any problem. So those were a few success stories. We also have problems, of course, and big problems. And me and my team, we are quite big supporters of the RNAi technique, and we expect solutions from that technique. And we hope that in the near future, we can start doing trials with it, so we can show our growers that there is something there. The first gap that we have is aphids. It's a really important one. It's really difficult to get rid of them, and they interrupt our IPM strategy quite drastically because we have to correct with spraying, and spraying is not selective, and you will hit our predatory mites as well. So for the aphids, we can use some biological predators like predatory wasps or ladybugs or chrysopa, but yeah, it's not sufficient. It's not consistent, and it will not prevent the problems that we face with aphids. Now our growers are trying to clean up before flowering with the spraying, but yeah, it's a really low success rate because most of the aphids will appear later in the cultivation. So we need to do corrective measures, and that's something that is not selective, and it will disrupt our IPM strategy. We use chemicals, they're scarce because of That's what Anna already told you. They're disappearing. And we have some biological sprayings that we can do, but we have to repeat them multiple times, and it's not very effective. So RNAi would be a very good solution for this because it's a selective spraying that we can do. It will fit perfectly in our IBM strategy, and it's not chemical either, so perfect. The second gap is root diseases, Phytophthora in strawberry. You see there a picture, complete loss of plants. The only solution we have there at the moment is chemical. We have to treat our plants right after planting, so only once, luckily only once, and during the plant production phase, but that's not where the fruits are being produced. But we do need a non-chemical solution because the products are disappearing, and yeah, especially during the plant production phase, we have to spray multiple times. It would be much better also for the Green Deal, of course, to have a non-chemical alternatives. There is an alternative now with the plant sauna. It's a treatment with temperature on the plant material to get rid of the disease. But we're far away from a very practical application of the technique because it's not possible to treat very large quantities of plant material. And that's needed. We're talking about millions of plants that are being planted at the same time. They have to be treated all at once, and yeah, it's not possible with the plant sauna at this moment. So there as well, RNAi can be a very nice addition as a tool for our growers. In the third gap is fruit rot diseases. It's already mentioned a few times. So in strawberry in Belgium, we are facing quite a lot of problems with botrytis and mucor. The current practice is spraying, of course. It's spraying both chemically and biologically. It's switching between them, but the efficacy is coming from the chemical products, not from the biological products. The problem with the chemicals is they're not always selective. Also, the predatory mites sometimes suffer from it. And for that reason, we need a non-chemical solution as well, which RNAi can be. And the last one I would like to mention is Drosophila Suzuki. We don't have really a good solution for this problem. So it's hygiene measures, so monitoring with traps. We do sodium testing of the fruits to see if the larvae are in the fruits. And then if it's necessary, we have a corrective spraying with chemicals, like spinosad is used quite often, but it's not selective for predatory mites either. So it's very common that we spray against Drosophila. The problem is solved, but 2 weeks later, we got a really big thrips problem because our predators were killed. So it's not really the way to go in the future. So RNAi can be a useful solution as well here. And then I would like to show you, because I talked quite a bit or mentioned quite a bit the side effects of chemicals. Here you see a slide of both fungicides and acaricides and their side effects on Amblymodus limonicus. Amblymodus limonicus are most important predatory mites against thrips and whitefly. And you see that a lot of the acaricides and insecticides do interfere with our population. Like products like Exalt, Raptol, Tracer, Tracer is spinosad, Vertimec is abamectin, Desis, Karate, they're quite toxic for our predators. So corrective measures with those products is just not a great idea for our growers. And they need alternatives for this. Also, the fungicides, they're not quite as harmless against the predatory mites. They do have an effect. Like Malvin is Captan. So you see some effect, quite a drastic effect on our predatory mites. And the last slide is the slide about residues on strawberry. You see that the sprayings against powdery mildew and botrytis at this moment are the main cause of residues on strawberry. The rest of them is not so pronounced. For powdery mildew, I mentioned already the UVC technique. That will be a very good solution for that. But for botrytis, yeah, we don't have the right answer yet. And probably RNAi can be a very helpful addition to get that percentage lower. And that was really short, my presentation about strawberries and RNAi. If you have questions, please ask.
Thank you, Peter. Any questions from the audience?
Well, it can open the discussion in some way. So
yes.
And I see an answer from Salvatore to Andreas regarding the rootstock. I fully agree. There are no dossier filed until now. And in reality, so we don't know. In fact, in my opinion, this is one of the main issues, the fact that scientists together with industry, with this type of organization should continue the research. And if there are positive results, we should work together in try to promote this work. And really, at this point, fill up the dossier and try to see if we can have some kind of clear benefit and results, and then also in terms of final approval and distribution, see if this may happen. As far as we know, the most advanced product in this technology is what we have seen this morning, presented by Nick. So the plum, the virus-resistant plum. And let's hope the new product will come out, in particular for fungi resistance. Botrytis work is progressing, and maybe in the next year, probably beside our work, because we are not the only group working on this. There are many groups working on fungi, in particular botrytis. So this can be maybe one of the first products that will be delivered or will be prepared. And so again, a file will be, a dossier will be presented and submitted. There is a question from Peter, from Kalle to Peter. Is there a statistical significant effect of fungicide on predators? Peter, do you see the question from Kalle?
Yeah, I do see the question. Yes, like the captain was significant. Yes, there was and also we tried with fluopiram. There was a significant reduction in their population of predatory mites. But it's not really a drastical something, because once a population goes down to, let's say, 60%, they can recover quite rapidly. So it's not that big of a problem as, let's say, an acaricide that will kill off 80% of the population. That will be really difficult to recover from. So that's another thing that is very important in this matter.
About your question, Peter, regarding the, for example, well, I think that for insect and diseases affecting strawberry, RNA can offer different possibilities, because are only fungi and insect. And regarding the application of RNA technology in protected plant from fungi and insects, there are really many studies already available. And in particular, for example, the case of Phytophthora, just to tell you, you mentioned the fact to spray. In reality, there are many studies now, yes, try to, actually that is a comment also coming from Jeremy. There are many studies related to the delivering system. So spray, it's an option. But absorption from the root, it looks like it's another option. It has been demonstrated that RNA can be absorbed from the roots. So again, the work has to be done first to identify sequence that can target gene for Phytophthora. And this is a work that is already existing in some way. I don't know, maybe Elena can give us some indication about that. And the question here is how to prepare formulates that can help this new sequence to remain stable and make it more easy to apply them in the fertirrigation system, for example. In particular, in your case, you are working a lot with solid S cultivation system, and this is clearly an option. Another, I want to remember to all that another delivering system for double-strand RNA is also drilling by injecting. So maybe for tree, not only is the solution of spraying, but probably for some diseases, another solution can be to inject the formulate with double-strand sequence directly in the axillematic or phloematic or in the vascular system. Probably this is another solution. So all these solutions are open and are based on identifying the most appropriate sequence, formulate, and delivering system. Other comment?
There's a question from Jeremy Sweet. Do you think RNAi could be applied in hydroponic nutrient solution?
Well, more or less, I was already giving it.
Okay. Okay. And Peter Mellis says that growers are very interested in techniques like RNAi because it is not chemical, highly selective, and can be sprayed. So he thinks that growers will make use of this product once approved. And here I would like to make a question for producers. So what's the producer perception about consumers? So the consumers are, let's say, the demand side, so they need to be satisfied and pleased. What's your perception about it? So do you think they are aware enough of the problem and the challenges you mentioned? Do they have the right knowledge to choose products? Do you see that shift towards more sustainable consumption models? Or do you think they are aware that sustainability needs innovation? So what's your perception?
Can I express my comment? But this is Mirko Montefiore's comment, not the producer or not the company's. This is my personal comment. I think that the consumer is not aware of the challenges that we have, and he has not to be. I mean, that is my job to give him the best product and the good product. The consumer wants to eat something that he believes to be safe. He wants to eat something that is good quality. He wants something that is, nice, good, tasty, and is available at this table. And of course, it's right to pretend that it's something that is safe. But the challenges that I face in the orchard to produce, I don't think the consumer will have to worry about it, also because he doesn't understand it, and I don't understand other field of how to make my mobile phone, how complicated it is, but I want it, and I want it performing and at the right price. And also for the techniques, how he has to understand what are the techniques that we use. I don't know. If I say that the asparagus that he eats, they are produced by double haploid, wouldn't he be scared? I think that he will be scared too. I think that is I don't know if he has to understand horticultural science is a science, which I think that is not the grower that does understand. it's not the consumer that has understand the horticultural science. He has to be sure that he eats something that is safe and good. But as I said, this is Mirko's opinion.
Thank you. Thank you, Mirko.
If I can add something, yes, I think that farmers know that consumers are requiring more and more sustainable products with low residues, with few treatments. But I think that one of key subjects in agriculture, in direct contact with farmers, in my opinion, are technical advisors. Because farmers, I think that they do not have so much knowledge about what could be prompted in the future, all the different kind of innovations. So I think that advisors should promote positively these kind of innovations, for sure.
Thank you.
I agree. So quality is the major issue. The consumers demand high quality of the product. Secondly, they want to be as healthy as possible, so the amount of pesticides should be as low as possible. But even with chemicals, that's possible if you use them wisely. Listen to your consultants. It is possible to produce fruits with a low amount of residues. And I think if we can both deliver quality and low residues, the consumer will be happy. Yeah.
Thank you, Peter. Bruno, in the chat, there are some comments.
We were interacting with Peter was corrected by Andreas regarding chemicals, the use of terms chemicals. And I was interacting saying that everything is chemicals, but I think that in webinar 3, where there was a discussion about the rules and the assessment of the new product, RNA-based product, it was clear the definition given by Mike, don't remember his name, from APA, United States, but in any place, RNA are nucleotides, and we are eating nucleotides, or as we were saying, are not protein, are not enzyme, are nucleotides, are a small sequence of nucleotides. So that we should keep in mind this, and as clearly Peter was saying, this small sequence of nucleotides have very specific target, and we can really understand, even expect from bioinformatic analysis what can be the risk of target effect. Clearly, then this can be also further proved with some kind of additional study to demonstrate that there are no unintended effect or risk of target effect from this small sequence of RNA.
Andreas is saying, Bruno, I agree with you. I am a bit skeptical on the use of the not chemical.
Yeah, I can agree, but again, it's a different chemical. Well, I don't know. Maybe we will discuss this after. The main issue for the new potential development of RNA-based product will be the classification. We already say that this will not be classified in the GMO because it's not altering the DNA of the plant. It will be classified as a new pesticide. And as far as I understand, maybe in the discussion we will have it in the afternoon, that we have people also from DG Sanko, we can ask and see how they can clarify this, that there is a difference between the chemical molecule, because there are the chemical pesticide, a specific regulation, and the biological pesticide that includes a huge range of molecular of different origins. And so we need to understand in reality, where this type of molecule will be included in the new regulation. But again, we don't know because at least at the European level, no application have been submitted. So as soon as we will have applications submitted, we will understand how this will be approved or if how it will be assessed and also classified. At the moment, as far as I know, there is not real a position or a decision at any level on the final classification of this product. Clearly, the final classification of this product, in my opinion, will have a very strong impact because depending to the final classification, then after you will see how far you can go in the utilization of this product only for some kind of agriculture or for some other kind of agriculture. But the main difference is between integrated agriculture and organic agriculture that make the difference and make clearly more selective in choosing accepting some kind of molecular to be applied for protecting plant.
There's a question from Peter. Do you think RNAi can be applied in organic farming like pyrethroids are now being used?
Well, this is, I don't know. In my opinion, it depends from the classification. I don't know if some people in the organic sector are still connected. I know that somebody was connected. It would be nice to know what is their opinion. From the chemical point of view, again, if it is just a question of chemical point of view, this has to be assessed and decided. I have, in my opinion, in Bruno's opinion, I think that this type of product could be helpful also for the organic production sector. But clearly, I'm working on this, so you can understand the reason of my opinion. It will be interesting to see what the sector is thinking about, and maybe we can discuss. And clearly, it will be the issue in the next period. But again, are there some comments regarding the question from Peter Melis?
Yeah, maybe I can quickly comment on it. I'm Martin Sommer here, and I work for the EFORM Organics Europe. I'm mainly here today to listen to what you have to say and yeah, to also get an overview of this new technology. So thank you very much for all the presentations. They've been quite interesting. Yeah, regarding the application organic farming, I mean, like Bruno already said, it all depends on the regulatory status, and then the question, like, is it compatible with the EU organic regulation, so the current one, but also the new organic regulation that will come into effect 1st of January 2022. Yeah, so that's a bit the question. And then also like Ms. Aldigretti has said, the consumers' expectations are also quite important. Consumers also have particular expectations towards organic, and yeah, I'm curious because I think you will also have a work package that will go in deeper into consumer reception, so that would be quite interesting to see. Thank you.
Thank you, Martin.
Thank you, Martin.
Vera, if there are no other questions, I think we can stop because we already spent 3 hours and we have another section in the afternoon, so maybe other questions will come up in the afternoon. We usually take a final virtual photo. So if you want to turn on your camera, it would be nice to have a picture from everybody. So let me see. Let me have it enlarge. Sorry, we are. One is always missing, but let me try to get this and let me wait for another. Just sorry, just a minute because I had a problem in saving it. Are you still there? Let me take another one. Okay. Thank you very much.
So I would like to say thank you once again. It's been a very nice session. I remind you that in the afternoon at 2:30 there will be the second session, the meeting with policymakers, and I hope that most of you will be connected and have a nice lunch.
Thank you very much.
Bye, thank you.
Thank you, bye-bye.
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 Thank you.